I like to continue the story that's been developing. I'm 77 years old. I've been at this since I was 20-something. And I come from a long line of farmers that were the pioneers, farmers of the Susquehanna Valley in Pennsylvania, southeast Pennsylvania. So it's kind of in my blood. And my grandfather had a farm when I was a kid. And that's what I love the most, is going up there on the weekends and with my grandfather. And it was the traditional farm with a dairy and an orchard and sheep and pigs and woodlot and crops and cash crops. So I know what it used to be. And I know what it is now. And as an organic certifier and as a consultant, I've seen literally thousands of farms. I did farmer-to-farmer work for USAID, where I'd go overseas and I work with growers. And my big focus, I went, or initially anyway, University of Delaware College of Agriculture, and I studied entomology and applied ecology. And I was really more interested in the applied ecology part. And because I'm looking at the big picture, and but that's a whole nother story how that happened. And Vietnam, which got in the way, and I don't want to go into that. But anyway, my work was mainly as a practitioner, as a consultant with farmers, where I had to solve problems. It wasn't an academic. It was the real world where these people's lives and their family and holding on to their farms was at stake. And they'd have problems, and my job was to solve them. And I had to do a lot of study and research in order to come up with answers using the scientific method. And so that's how this all starts. So today, we have a company in Oregon, and that company it produces the products which I came up with over the years to work with crop decline, what we call crop decline, maximum economic yields, like what is the missing factors, what are the limiting factors of any given crop. And then this is why Dr. Dijkstra's talk was so good, and it really, it's a good thing to follow up on, because he hit on a lot of stuff that I don't have time to talk about. But he was right on. So I'm going to go and show you how that works together and how. Okay, this is how it works. Okay, this is back in 1973. I had a piece of ground in Montana. It was hard as rock. It was overgrazed. It hardly supported anything. And so I had the motivation, like, what can I do about this ground? So I saw a little tiny ad in the Montana Farmer Stockman, and it talked about something called clodbuster. What's this stuff? Something called humate. I never heard of it. I went to ag college. I went to a bunch of colleges. I never heard about humate. What's humate? But I tried it, so I didn't even know how much to use. So I just took some sweet corn seeds, some of the ground from my burned out ranch, planted some corn seed, put a pinch in each pot, and then I put them in my little geodesic dome greenhouse that I built and watered them with rainwater and see what happens. And I'll say, that stuff works. So this wasn't about academic or studies or anything. It was like, let's try something. Wow, it works. Didn't have a clue. Why? Did not have a clue. Okay, so this is many, many years later in Oregon, when we did have a clue, right? And we came up with a concentrated way of activating him. I'm talking, this took 50 years, okay? And so then I took, from my latest farm in the Willamette Valley of Idaho, Oregon, which is really good soil, took samples from the field that had been nothing but fescue grass seed for like 20 or 30 years. Nothing but nitrate fertilization. No manure, no compost, no nothing. Dead soil. The farmer eventually went belly up, okay? So I put some of our products on and just got some bird seed. You know the kind you put in a bird feeder? It's a mix. So you got some sunflower, got some millet, you know. And I just put our different products in the control groups, and wow, this really works for bringing back, you know, declining crops, burned out soil, or whatever you want to call it, dead soil. And so that's the beginning, and I won't say this is the end, but this is like 50 years later. And we've perfected, instead of using a dusty, dry, messy stuff from New Mexico that you have to use 200 to 300 pounds an acre, we're down to something that is dry, soluble, not dusty, and you only need to use at the most 50 pounds an acre. Okay, so now I'm going to go back to, now we're talking about what Dr. Dijkstra was talking about. Okay, and I didn't know why. Okay, so as a consultant, in mainly IPM, soils, agronomy, this is seed potatoes. I worked with the seed industry for many, many years. The seed potato industry in Montana, and we're very concerned about diseases and insects. It has to be certified disease-free, viruses, etc. So seed potato production. So the growers are saying, you know, my crop hasn't been so good lately. You know, we're not getting a grade out and one thing or another. I said, well, look, let's try some of this humate stuff. I did some little experiments and it really works. So let's buy a load from New Mexico. You know, it's only 20 bucks a ton. So let's get a load. Okay, we'll get a load. And so we stripped a 40-acre field with and without. And you can see the difference on the right there. Well, it says with 250 pounds. Was treated. This was just broadcast, disked in, and then the potato crop was planted. In seed potatoes, you don't plant until June because you don't want big potatoes. You want little. And so on the right next to it, you see less vigor. It didn't close the rows. That's something they really look at. But here's the fun part that got me started on this whole thing. The potatoes on the right that were treated with humates had no leaf damage, had no insect damage, had no insects at all. On the other side, without the humates, Lepidopterus defoliators, other insects, were eating the leaf to where it was like lace, holes that we call Lepidopterus defoliators, you know, caterpillars and other things. Okay, so here I am, a young guy in his 20s, and I want to save the world, you know, organics and biodynamics and all this, and I see something like that. My field in entomology wasn't like spray and pray. It was biocontrol and, you know, natural methods of control. And so what I saw was, it started me on the track. I mean, what's causing this? And of course, we did some tests, and yeah, the Brix was up on the treated side. And then over the years, you know, that was always something that growers that would take a look at it would notice, you know. You don't have to take a Brix reading on a lot of this stuff, just taste it. Or you'd put the humic substances on a pasture. And I used to raise livestock, sheep, cattle, and even pigs, and I raise chickens now. So so anyway, so you'd put the stuff on the ground. All the sheep are over here. They're not over here where we didn't put it. They're over here where we put it. And they'd eat it right down to the row, right down to the control, preferentially, the treated areas. Well, the sheep could tell it's sweeter, right? And maybe something else. But of course, they love sweet stuff. If you want to make sheep come, you have some molasses added to your grain, and they get a whiff of that, and you can lead them right off a cliff. So they like sweet. So that's another thing we we noticed the unexpected effect, unexpected from trying stuff, without anybody influencing me to say, that's crazy. I'm my own master, I was then. I'm not an academic, I had nothing to do with that. It was what decisions I made and the farmer made. And we'd find stuff out by trying stuff, just like our ancestors did. But I mean, it started with, the usual scientific method. Well, let's do an experiment. Okay, so the big problem in Montana, and the reason I'm talking about Montana a lot here, because it's similar to the conditions in Alberta. And I also work with farmers in Lethbridge, Alberta, so I'm somewhat familiar with. So they were complaining, the growers were saying, well, we have problems with phosphate availability, and the fertilizer dealers just tell us to put 200 units on per year. And I said, well, why are they saying that? Well, here they give us a free soil test, and it's showing like 5 or 6 ppm. We have low available phosphate. I said, let's try another test. So we do P1 and P2. Well, yeah, your P1's low, but your P2's high. And then I said, well, let's do another test. And I work with seed growers, and they're a lot more, they want high germ, and they were having problems. And they were saying, well, we got to put more phosphate in. And I said, let's just see something here. Let's test total phosphate. Let's go a step farther. 10,000 pounds an acre. I said, look, you're not going to have to add phosphate for the rest of your life and your son's and family's life, okay? That's not the problem. So I started looking into it, research from all around the world, and it turns out that humic substances, now this shouldn't be a big surprise, humic substances are crucial to phosphate availability. And again, we didn't know exactly why that was, but you could demonstrate it. You could easily demonstrate that. And so we started using phosphate. I made a product called Humiphos. It was a combination of rock phosphate and humic substances. But then we found out that on most soils, that's not what we needed. All we needed was the humic acids. And the phosphate levels, and I'm talking about, we're monitoring everything. Soil tests, leaf tests, petiole testing, extensive monitoring. So we knew what was going on. So we'd see increase in calcium, we'd see increase in phosphate. We didn't apply any. Now this is an alkaline soil. It's not like Willamette Valley, but so that's what we were doing. That's where it all came from. And so phosphate availability, we found, is affected by, for one thing, mycorrhizal fungi are very crucial in the release of insoluble bound phosphate, and the plant gets some phosphate, and the mycorrhizal gets some sugar, so it can maintain its life cycle, and it produces spores, and we go from there. The other things that was interesting, some research from all over the world, and indicated that in some places they test for silica, because there's no point in applying phosphate if your silica levels are low, if your silicic acid. So the first thing they'd want to handle was the silicic acid issue before they would apply phosphate, because they didn't get a good response if they had low silica. Sulfur, wow. That became a big factor where we were working in the Flathead Valley in Montana and Washington State and even in Canada. These people are suffering severe sulfur deficiencies. They're worried about phosphate, but boy, they got a bad sulfur deficiency. And you'd put sulfur products on. We had one that was 13% silicic acid, pH of 2, and about 25% sulfur. This was mined in Utah from a volcano, right? You'd put that stuff on, it was like magic. You could see right to the row where you put that. Alfalfa yields, wheat yields, everything. So we were putting something on there that is a combination of sulfur, very available, silica in an acidic form, and we also had high pH soils. So we were reducing the pH of the soil, adding the limiting factors, and then also using humic substances along with that. Wow, then you get up to record yields. Now that's mycorrhizal fungi. They live on the surface and they penetrate, let's say they penetrate the root, and they have nodules on them. They have little pads, little cells in them that hold water. So not only do they produce enzymes that releases phosphates, they're a biocontrol agent too against plant diseases. Like I found out in my later research that a lot of the decline we were having, root rot in beans and other crops that I was working with, were like weak, opportunistic fungi, like Phytophthora, Rhizoctonia, on and on and on. Well, the idea, and a lot of these were developed at Oregon State University, the idea of mycorrhizae as a biocontrol, because it resists all those weak, unspecified fungal infections of roots. But it also, not only does it protect, it feeds. It feeds, and it has little nodules, let's say, vesicles. It look like a little tiny plastic bag under a microscope that hold water. And so when you get a drought situation, the plant can feed off of that stored water. I mean, this is a true symbiosis. And people wonder, like, why is my crop going down? Why don't they talk about dead soil and all that? Well, yeah, these microbes and the mycorrhizal fungi and all the other things can't function. You've killed them off with salt fertilizers, with pesticides. I did a big job in Hawaii. Why did the macadamia nut decline? Why are these trees dying? Why aren't we getting good yield? It's interesting. They use Roundup to spray the ground to get rid of all weeds, all vegetation, to make harvesting easier. Okay, well, there's little vesicles on a macadamia root. They're like little brushes that are really close to the surface that scavenge phosphates from the breakdown of leaves and organic matter in a rainforest situation. Okay, let's spray Roundup. Well, what's Roundup? It's glyphosate. They make it out of phosphate rock down in Pocatello, Idaho, where they make phosphoric acid first, and then next door at Monsanto, they make the Roundup, right? Okay, they're spraying this on these plants, and they're going down the tubes. So their management method using Roundup was destroying 3,000 acres of mac nuts. And of course, nothing can function. I turned that around, but only on a small scale because, the fertilizer salesmen and the chemical guys were getting kickback. Hawaii's kind of corrupt. So we couldn't really do much because, it requires bribery, and it's a weird situation. So humates, we also found by extensive testing, that, and this didn't make sense either. A lot of this didn't make sense, but it worked, and that's all that I cared, and the farmers cared. But I found out many years later, humates are amphoteric substances. They buffer the soil. It can either buffer it up or down. So if you have a very alkaline soil, pH tends to come down. If you have an acid soil, pH tends to buffer up. And then we use the Albrecht method, soil testing, so we know the saturation of the cations, and we test for trace elements. And then we now use a soil food web microbial analysis, throw that in, so we see what the distribution of the microbes is. And it's really, what's important is the distribution of the trace elements in the right ratio, along with the cations in the right ratio, and the microbes in the right ratio. That means fungal dominance versus bacterial dominance. So when a crop or field goes down, it's because it's becoming micro or bacterial dominant. And I did a paper on this. It was presented at the International Humic Substance Society in Japan. And using our, in the soil in the Willamette Valley, which you find plenty of burned out soils. So it's a perfect place to do that kind of testing. So we recommend along with trace elements, sulfur and silica, must be considered. And then testing, a good soil audit is a starting point. Okay, so now we call humic acids, it's changed, everything's changed. Now it's like they're getting acceptable. They wanted me to do a presentation to the American Society of Agronomy in Tampa, Florida, one of their meetings, their scientific conference on humic substances workshop. But years ago when I'm in my 20s, snake oil peddler, all that, right? Now the American Society of Agronomy wants me to do a presentation. So that's how it's changed. So humic acids are biostimulants nowadays. They're complex mixtures of compounds produced by soil fungi and essential to plant health and growth, and not plant health, that's misleading, cellular health. This is a cellular phenomenon. It's not about plants. People will say, well, what will this work on? Will it work with animals or all my plants? If it's got cells. Okay, so we got into the animal health side with subsidies from the government of the Netherlands, because you don't get that here. Organic matter must be converted to humus and humic acids to be available for soil microbes and plant uptake. So there's a series of microbes involved here. And it's very complex. Okay, so we go to this is all about humus. Think humus, think soil tilth, think the smell of forest soil, the feel of a good rich soil, the color, black versus white. Which soil would you like to have? A white soil or a black soil? Okay, the Latin word for human is homo, also derived from humus, the Latin for earth. We're totally dependent on this formula here. I mean, we are the soil. Once the soil's gone, degraded, that's a symbiosis we have. We're not going to survive. And many civilizations, of course, played out their soil. There was climate change. There was oxidation of the organic matter, like nowadays in a massive scale, addition of salt fertilizers, et cetera, and those civilizations vanished. So we talk about history going back 3,000 to 4,000 years. No. People just like us were around 80,000 years ago. So what happened between the 3,500 and the 80,000? Now this is debatable because not long ago, they were saying, well, everybody came over the land bridge, blah, blah, blah. Well, now we found like fossil remains in a cave in Oregon that refute that. And now we're also finding civilizations like Göbekli Tepe that also refute that whole scenario of that go back, way back. So people on this earth have been growing foods for a long time, and they managed to maintain big, and that's the kind of stuff I'll be talking about tomorrow with agroecology discussion. So I don't want to get carried away on that. So where does humus come from? Well, humus is the source of humic acids. Humic acid is not a single compound. It's 800 or more from 3 fractions. So humic acids with an S is about molecular weight. There's no such thing as humic acid. It's not a chemical. It's not a compound. It's a fraction. It's one of the few things in chemistry that's defined as a fraction. So you have humus, and you can fractionate that with chemical compounds, alkalins and acids. And humic acid is defined as the fraction of humus or humic acids that's alkaline soluble, okay? And then the other end of the extreme is fulvic acid, which is defined as the acid soluble fraction. It's soluble in alkali, it's soluble in acid. Humic acid is only soluble in alkaline solution. Okay, and it's about molecular weight. The humic acids have a high molecular weight, and the color is going to be somewhere between gray and brown. Black is not humic acid. You're out of the range of humic acid. Black ain't humic acid. There's things in humic acid, or like leonardite, lignite, even compost, that's black. That ain't humic acid. The darker stuff is too high molecular weight to be absorbed by plants. It has to be broken down by microbes. So the humic acids feeds the soil, the fulvic acid feeds the plant, because it can be taken up, it can be translocated through the cells, and even through your skin, even as a foliar. But the humic acid, no. But a lot of humic acids, like our full humic, also are very high in fulvic acid. So that's why you get all the both effects, the feeding the soil and feeding the plant. So they have different structures, and they affect soil structure and moisture holding ability of the soil. And they have, the fulvic acid has antibiotic effects and antiviral effects. So humic acid degrade over time. So you're talking about something, if you put it in the soil, it's only going to last 60 days. But it kicks off a cycle of the soil food web where the soil becomes more fungal dominant, because it's a missing link in terms of fungal growth versus bacterial growth. And yeah, then you have all these effects that have been studied all over the world. The anti-drought and frost effects, the hardening of the soil, you know. So humic and fulvic acids degrade, and then once they degrade, yields are reduced, and crops are now more susceptible to insects, diseases, drought, and frost. Fertilizers do not make up for this loss. The soil hardens, more runoff, more soil loss. Time to move on to a new land or go to town and get a job. This is no joke. I can tell you because we only have 1% farmers now, so a lot of them moved to town and got a job, apparently. Time to move on, okay. Well, back to, so compost, yeah, you don't need to buy any humate or humic acid. You can make your own humic acids by composting. But we found that some of the growers we work with, they're just too big, and the availability of compost, the hauling it, the spreading it, it was just incredibly, expensive proposition and logistically difficult. So it's kind of impractical in a lot of cases to make your own humic substances. It takes 12 to 15 tons of compost. I'm talking on a wet basis, as used. Compost is going to be over 20% moisture, probably more. So is equal to the humic acids in 1 kilogram of full humics, because that's over 50% humic acid. And compost are down there in sub, numbers. So that was work done at UC Riverside in California that come up with that number. That wasn't me. Okay, 12 to 15 tons of compost equal 1 kilogram of activated humic substances. Okay, the humate acceptance cycle, or why, this is the paper that I gave to American Society of Agronomy, okay? Humic substances hold promise, help grow and sustain agricultural productivity, but what happens is you get a company comes in with a product that works and gets good results, and there's some research done, maybe by universities, and yeah, we're seeing something, and that encourages more use. People try it, and then companies jump in with anything that's black. It's humic acid, because we didn't have a good criteria for what's doing it. What's the test method? How can we determine? And it turns out that all these humate deposits are different. And since then, we've done all kinds of plant bioassays, functional group analysis, Fourier transform spectrophotometry, all kinds of high-tech things. Now we know. Now we can look at a sample of something black and tell if it has what it takes out of those 800 different compounds, like which functional groups give us the biostimulation. And it varies. I've tested at least 12 different ones just from the US and Canada. And they're all different. And also me and Rich Lamar, who's the guy who developed the test method. It's in his name, the Lamar test method, which is now used by, to standardize the testing of guarantee analysis of humic substances. But what would happen, these people would come along with these, we don't know what it is, but it's black. It tests high in humic acid, which is a simple test, humic acid. I mean, coffee has humic acid. You just get a cup of coffee, dry it, and weigh it. Humic acid. It's not going to do much. So what happens is it gets a bad name. So people are putting out products that don't work, and then farmers and researchers are saying, well, see, humic acids are snake oil. They don't do anything. And then the cycle starts over again. It takes another 20 years. And so I've seen this cycle repeat. And now we have a certified test method. That's that little green triangle at the bottom of the Humic Product Trade Association. So that's getting closer, but there's more. This is what's really going on with humic substances. And like I say, they're defined as fractions of humic, and they need activation, and they convert them to a potassium salt of the humic acid. That's using potassium hydroxide to activate and release the compounds. Now, our product, Full Power, is a pure fulvic. It's used for human, animal, and soil application. It's not made by using a hydroxide. It's a fermentation by microbes. And since it's fulvic acid, that means it's got to have a pH that's acid. There's people out here selling fulvic acid with a pH of 8, 9. Acid? No. Fulvic acid is a pH of 3, 3.6. Okay, so what we're talking about is not simple. So using nuclear magnetic resonance testing of humic substances, and they're the different fractions, humic, fulvic, olmic, and human, and Humin is insoluble, we get these different functional groups, your ketone, carbonyl groups, your quinones, carboxyls, your phenyl carboxyls and aminos, your aromatic and heterocyclics, you've got your aromatic protonated carbons, and then class E, methyl and methylene. Okay, methyl and methylene are toxic. So to get a score for a humic acid, you add up the positive functional groups and you subtract the negative functional groups, which is the methylene. That's sulfur-derived kind of thing. Now, these functional groups translate into all these other organic compounds, and we're talking about species of carbon. It's not just carbon that's important, it's species of carbon. This is organic chemistry. So it's about carbon, oxygen, and hydrogen. And all the drugs you take, the way your body works is biochemistry. And so you need these various functional groups represent things like flavonoids, you've heard of polyphenols, that's crucial. So you see that pathway? Polyphenols, tannins, okay? So the work we did in Germany at Humboldt University of Berlin using molecular biology and doing very complex testing, and I didn't learn in college, it didn't exist. So that's pathways, I'll get to that. And those pathways, once they're opened, and it requires these polyphenols and tannins, it's like throwing a switch, and then the plant has resistance to insects and diseases. And again, I mean, this could be directly related to the sugars too, but we've identified, and that's part of it, I'm sure. But the other part is the cell, or the plant or animal cell, has a mechanism. And that mechanism is activated by polyphenols, tannins, and other things that turn on its own resistance to pests and diseases. Natural resistance, just like we have, or we'd all be dead. If plants didn't have natural resistance, somehow they'd all be dead. So it's quite complex. So, you know, the idea that humic acid is like some simple thing, humic and fulvic acid, no, it's not. Using this, well, it's a technique of analytical biochemistry where we can see spots on different functional group, like a pie chart, functional groups, and we see dots on there. Each one of those dots is a compound. Now, we haven't identified all the compounds. There's 800 of them. So we go by functional groups, because we know which functional groups contain the bioactive components that we're looking for. It's mainly the aromatics. So we need, I don't want to get into biochemistry too much here, but just take my word for it, we know. And it's in the literature, at least in German literature. Okay, so this is how I do it, because I didn't have a fancy lab. I have a lab. I have one of the best labs on the West Coast to actually test by the test method. That's a certified method to test for these things. But we use seed soak bioassay, where we soak the seeds in different concentrations of humic substances, mainly fulvic acid, because it's got a low enough molecular weight, it'll actually get into the cell. The seed is one cell. Okay, so when we get that one cell to divide in an optimum way, it's the model for the rest of the plant. So it has to start with that seed. And so we can determine by soaking seeds in solutions, different concentrations of different humate sources. And what we see is in root length percentage. In other words, we see speed of germination and we see root length. And that's what we call the root length bioassay. So if it's really got fulvic acid in it, you're going to see a difference between the control and the treated. It's going to germinate quicker, and it's going to have a longer root system, a longer initial root, and then later on, completely modified. A fibrous, what I call a fibrous root, very, lots of root hairs, versus what I call stringy root, which doesn't have all those root hairs and it's not as extensive. And that all starts with fulvic acid is the only thing, maybe somebody can challenge me on this, but it's the only substance that will give you these kind of effects. And that includes synthetics, as far as I know. I mean, maybe someone's, but this is, after we do all that fancy research with the functional groups and all that to screen out the whatever, we go to this plant-based testing. And we see you can see what the optimum level is, 100 ppm or milligrams per liter. That's important because you go beyond that, it goes the other way. So you got to know exactly how much, see? So there's a specific number that concentration needs to be. You go over, it takes it the other way. It's a hermetic substance. It's either a medicine or a poison. Depends on the dose, okay? And so you can go worldwide, and there's research all over the world on humic substances from Russia, who was pretty big in it, all the way through Europe, the Middle East. The Chinese are really ahead of the curve because they used humic substances as medicines dating back to the Ming Dynasty. It's in their medical literature. So they go a long way. But like I say here, this is pretty well accepted worldwide. It chelates soil nutrients, improves nutrient uptake, phosphorus, sulfur, nitrogen, reduces the need for nitrogen by about 20%, 25%, removes toxins. We use it to reclaim mine sites, to complex heavy metals, PCBs, things like that. And it stimulates soil biological activity, which we can measure. And then it solubilizes minerals, including rocks. And farmers used to ask me, well, but you like kind of a snide, like, oh, can it solubilize rocks? Yeah. If the parent material is high in potash, yeah, it can release phosphate from rocks. Yeah. Sounds crazy, but that's how it works. I lived in Hawaii, high canopy rainforest, with 150-foot trees loaded with all kinds of vegetation. The parent material was recent volcanation. In other words, lava flows that were 2,000 degrees centigrade. Now you got a high canopy rainforest. Nobody did a thing. What happened? Okay. Beware. All humates are not the same. Don't get caught up in that, because you're just throwing away a lot of opportunity, a lot of good things, because of that bad experience that you're going to have. And I hate that. It's been going on for, like I say, you know, I started out in '73. I probably see that cycle 3 times. So we're kind of there, and our products are actually registered and have a guaranteed analysis. Aha! You know, most don't. And also we're registered in Canada and the US. Why does soil organic matter? Well, it's everything. It's the difference between the earth and the moon. That's how it matters. And you saw that. The word human, humus, all that, that's us. That's us. Once the organic matter is gone, well, we're gone. And then you see the inputs is soil, roots, and tools, and the outcome, soils, plants, and animals. And this goes back to the Stone Age. Okay, so farmer belief systems are a factor in this. Back in most of our ancestors' times, and I'm talking now specifically about the Frisians, but this also applies to other groups in history. They'd have a bad crop, there'd be problems with the crop, they're all upset, what are we going to do? Well, we had to sacrifice a girl to Odin or some other entity, and we'll get good crops again. So that's what they're doing, see. So believing sort of superstition, you know, they really, you know, didn't know how to answer the problem. Like the Romans had problems with powdery mildew because they grew a lot of grapes. Powdery mildew is still a problem on grapes. There's a god for powdery mildew, Rohibus. I probably pronounced that wrong. There's a god for powdery mildew, and you make sacrifices and offerings to this god to get rid of powdery mildew. That's what I'm saying. The only thing that's going to help us is the right kind of science, true science without any biases. The principles of biological farming have evolved for a century, and now advanced biological science and microbiology. We are now learning faster than ever that microbial activity and concepts have scientific backing for results, economics, and sustainability. Salt fertilizers and pesticide-based farming is unhealthy for the farmer and family as well as the world itself. Based on what we know, not scientifically applied, more of a marketing bias promoted by doctor experts at the state universities. Okay, funded by big petrochemical money. The rest of the world is also working on this and freely offers to the world, and I study it and advocate for farmers who want to see a change and keep their kids in farming. I tell you the truth based on the best knowledge available on how to achieve the maximum economic yields. Lower cost per unit and sustainable with a committed farmer. So you got to adjust your thinking a little bit. And I don't take any pot shots at land-grant colleges and doctor experts, because that's a broad generalization, because I get a lot of my information from those sources in published papers. But in general, that's why I dropped out of that system and went on to microbiology and general biology, and I didn't actually take a degree in entomology because I didn't want to. I didn't want to be identified because I went to a university that was funded a lot by the DuPont company. And I wanted to learn biocontrol, but no, that's one course, guy, you know. We're not. So my degree is actually in biological science, my undergraduate liberal studies. So that's everything. I took every ology in the university because I wanted to stay in the university as long as possible to avoid Vietnam. Okay. I did finally get drafted, but that's another story. Okay, so, okay, wrong way, huh? Uh-oh, fix it. Okay, so I did a lot of work with clotting soil. It's a big problem. Turns into cement and cracks in the summer. Turns into mud that'll you get stuck in. And I didn't know whether this is going to work, you know. But what we did in the greenhouse again, we took soil from the field, really bad soil, and put it in pots, and we treated it with fulvic acid, humic acid, and control, and watered it with rainwater. So there's no bias from groundwater. And after 60 days, we dumped out the soil and took a look at it, but we went past that. We did a soil food web analysis from 2 different labs. So it wasn't just one lab, it was 2 different labs. And then this is the kind of fungal activity you'll see in a fungal dominant soil. But what it showed was, well, friable soil. It's amazing. It's just one of those aha moments, really, for me and the lab, that it worked that well, that quick, with such a low concentration. And then, of course, what the result was reverse of dominance with a high level of fungal dominance after these treatments. And, okay, let's go back to sugars. This is research done by the Chinese Drought and Frost Remediation Branch of the Chinese Agriculture Ministry in cooperation with me. And they're very cooperative. Don't think they weren't. They're looking for a solution to problems. And we gave them some product, and they did a bunch of tests in northeastern China. It's kind of a dryland area like here. It's kind of cold. And they used it 100 grams with a seed at planting. And they got wheat yields increased 17%. You're starting at a low number, though, see. And millet would really respond. You got a 68% increase on millet, corn and buckwheat, and wheat yield. So this is the kind of yield increase we got, the Chinese got, just using a small amount with the seed at planting. Nothing more. Okay, so back to the sugars. Okay, so what we found out, and not just us, the Chinese, there's research in the literature from the US and Canada on the effect of humic substances on drought effects on wheat. Okay, so there's lots of data behind it, that it reduces drought stress, etc. Okay, so you can prove that yourself. You just take some wheat seed, put the soil in a tray, and have a control and treat it. And then I just take a bucket of water, wash the roots off, mount it on a board here, and let it wilt. Okay, so the wheat treated with the full humic or the full power, which is fulvic, there it is. I mean, the leaves don't curl. I mean, that's sitting out on a board with the roots exposed. And the control, it's rolled up. The leaf has rolled up. And the root system is not as massive or fibrous. So, I mean, that's a simple test anybody can do. You know, you don't need doctor experts or even me. You can do that yourself. You can prove it. The plants will tell you. Does it matter why? Okay, but here's why. Okay, here's the effects on wheat. This is, they gave me a book, the Chinese, because when I cooperated with them, they gave me a book with 7 years of research on humic substances, mainly fulvic acid, not just for plants, but also human health. They gave me the book. It was written in Chinese. I had to find a Chinese chemist to translate the book for me. So what they showed was the agricultural application of fulvic acid, Fulvic acid, an effective and easy to use anti-drought agent. Research on the use of #1 anti-drought foliar spray agent. Now they have a product in China, #1 anti-drought foliar spray agent. That's the name in Chinese. The other one is Dryland Dragon. Okay. The other one is #1 grape sweetening agent. Think about that. And it's fulvic acid, and it's sold to increase sugars, because the drought effect is like putting propylene glycol, or let's say even sugar, in your radiator, and that's going to protect it against freezing. It's not going to freeze at such a, it's going to change the temperature of the freezing. So you have a high sugars in these leaves, and that protects partially. I mean, I don't know whether that's the full effect or not, but it protects against the frost and drought by what I showed you. And they're not curling. There's high sugars, and you can measure it. Okay, so they did all kinds of stuff. A study of prevention of drought damage in wheat production, the hexicard using fulvic acid. A study of fulvic acid agent disease prevention and increase in wheat production south of the Yangtze River. Experimental results of fulvic acid spray on wheat production. The effect of water preserving and anti-drought agent on the growth and yield of wheat. The effect of fulvic acid spray on the increase of wheat production during the non-drought period. Experimental demonstration expense and use of anti-evaporation agent on farm products. The effect of #1 foliar anti-drought spray on the increased yield of wheat production. The effect of mixing 1 pound anti-drought agent with seeds on the increase of maize yield. Optimum rate showed 10% yield increase. This is China. They have 1.2 or 1.5 billion people. They've been farming for centuries and eons. They're trying to solve problems. They're trying to keep their agriculture going and feed all those people. They don't have any bias. They don't have any agenda other than that. And then in US studies, this is some people that are in the International Humic Substance Society that published this paper, but it boils down to demonstrated the combination of dry calcium with oxidized lignite, leonardite, combined to alfalfa, performed as well as a combination of calcium chloride and EDTA, popular synthetic chelating agent. So that's on calcium availability. And then a thorough review of the literature by Chen, who is a famous researcher in our world, concluded that humic substances at an application rate of 67.5 kilograms per hectare, which is about 60 pounds, and foliar application of humic and fulvic acid at 375 grams will affect plant growth. So there's the number. There's the number. So what we call it is priming the food web to improve tilth. So once you apply the humic substances, the right ones, you get the fungal dominance on a really massive, really rapid basis. And it's producing organic matter. We'd see a 2% increase in organic matter too after this first or second year. Where's that coming from? Well, it's coming from the increased root growth, the decomposition of the roots. It's coming from the fungal biomass itself. So you're increasing the organic matter. How does a soil go from sand or rock to a rich humus histosol, which is an organic derived soil? Where did that come from? The key is to establish fungal dominance of a virgin soil. Now, when I was working with the Mayan Indians in Belize and Guatemala, I observed, because I'll be talking about this in the agroecology approach, how they farm corn. And they got results as good as any modern corn farmer, but it was 2 years of corn and 20 years of brush. So they'd burn an area, they'd plant corn, they'd get 2 great crops, abandon it for 20 years, come back, plant corn again. If they waited 10 years, they got half a yield. So you can make humic substances that way. You can get fungal dominance back that way. You just, take 1 or 2 crops and let it go for 20 years. Now, obviously, that's not going to work in our society. So what we do is we prime the pump, prime the earth pump, you might say, and it'll just keep going for a while, you know, and you monitor it. It's creating its own humic substances. And of course, I'm assuming you're managing the decay systems, and you're incorporating crop residues and hopefully other organic matter inputs. This is the work we did. It's hard to see, but it's fungal versus bacterial dominance. The one in the middle is the control. You see, you have about equal fungal and bacterial dominance, and then at different rates of the different products. You know, the green is the fungal and the white is the bacterial. We get these big jumps in fungal dominance. And this is like a blend of two different competing labs. And then the soil improvement factors at these different rates. And believe me, rates are crucial to this. There's a mycorrhizal fungi under electron microscope, some kind of. So the principles are important. The soil food web, which is primarily fungal and bacterial, need an energy food source. They then fuel microbial growth, that in turn feeds the plant roots. Simplistic, but it is. The fungi and bacteria live around and in the roots and move ahead of the roots, converting soil minerals, elements to soluble forms the roots can take up. Now, who's our friend from Rutgers? Dr. James White. Dr. James White, Rutgers University. It's incredible the work he's done, but it's showing bacteria that are part of the root system. They're in the root. And then the plant needs nutrients. Those bacteria go out of the root, ahead of the root, and solubilize nutrients, or let's say ingest nutrients, and then they return back inside of the root and feed the plant. Ta-da! Incredible revelation there. And we work a little bit with him because it's just, I mean, some of these beneficial fungi we work with that are rhizosphere positive fungi, they don't work alone. They have like a bacterial association with them. It's more complicated than we originally thought. And then soil compaction is the #1 yield limiting factor. It's the one thing behind like the failure of civilizations is this compaction issue. And then here's some of the basis for that, you know, is your release and your nutrients. And, you know, you guys can look at this later on the YouTube or something. So, fix that. So this goes back, and I'll probably be talking more about that, to biodynamics. I was influenced by biodynamics in Pennsylvania. I'm from Southeast Pennsylvania, and there's Dr. Pfeiffer, and it goes back to the whole concept of biodynamics, which came out of Switzerland, okay? A man named... No, way before that. Steiner. Yeah, Rudolf Steiner. He was a genius in a lot of things. And he's the guy that came up and did the big lecture in Basel, Switzerland on biodynamic farming. Okay? And since then, the Swiss government has tested for 21 years comparing these methods, biodynamic, organic, and conventional. And the results are phenomenal. It was published in Nature, but then there's a lot of pushback. Oh, we don't want that, no, get it out of here. We don't want you to hear that. Because the biodynamic produced the lowest cost per input, the highest economic gain to the farmer. We don't, no. So nowadays we have the new sciences of molecular biology and biochemistry along with large-scale agricultural research over the last decade to provide answers. And so all this new technology, which I didn't know anything about, they didn't teach molecular genetics and molecular biology when I went to school. I'm learning it now with younger colleagues, right? But based on all that, we can prove the validity of biodynamics because we didn't have the tools before. The biodynamic community knew it, but couldn't explain it. So people were saying, well, biodynamics is kind of witchcraft, it's kind of woo-woo, it's kind of No. Yeah, because they didn't understand it. It's just like if I pulled out a cell phone, 200 years ago, they for sure would burn me at the stake. Okay, that's magic. Matter of fact, my ancestors were involved in printing the first Bibles. They financed Gutenberg, and then they took a load of Bibles to sell in Paris, and they had these quotes of Jesus in red. And guess what? My ancestor got thrown in prison. He said, well, it takes a year to transcribe a Bible, and you got a whole cartload. You got to be in league with the devil. And that's where the whole Faust legend starts. So he ended up in jail. So that's what I'm saying. What before was considered whatever, in league with the devil or whatever, condemned as snake oil. Next time I hear that, I'm going to deck the person that says it. Then science catches up. The tools always catch up. And so now we know. Now we call biostimulants and not snake oil. Like all this stuff I used to work with, the kelp extracts, the biodynamic preparations, the humic substances, all that stuff, man, snake oil. Now it's biostimulants. The US government's going to set up this program for it. Yeah, so I had to wait quite a few years. I'm 77 years old. I started this when I was like 20. Now it's biostimulants. The bottom line, basics for soil management to be sustainable, remineralization, the goal of organic biodynamic agriculture cannot be substituted for its basis of soil fertility. The addition of organic matter, liming, trying to get, if necessary, to obtain the optimum pH, calcium saturation, stimulation of the food web, use of rock dusts, and I did a lot of work with blended rock dusts based on glacial minerals and are needed in many cases, and humic acid should always be added to that program for rapid effects. I recommend soil testing, both for microbial levels and distribution, soil food web, along with the Albrecht method to determine mineral pH, cation saturation, and using our microbial testing protocols, we can look at the entire picture and come up with a plan for biorational management of soils on your farm. The goal of Faust Bio-Ag is to provide alternatives to toxic technology. That's been my saying for 60 years or so, something like that. I used to publish in Acres USA. I was at the first conference they ever had. My mentors were the guys that are passed on, that were luminaries, including Dr. Callahan and others that you might have heard of. And I'm the next generation. So I'm hoping that you guys can carry this on, because that's what it takes. The maximum genetic potential by advanced nutritional science and biological technologies that feature a full line of biostimulant products and reverse crop decline and correct all crop limiting factors. That's what I do. I work with the seed industry. I had to. They came to me with, oh, our bean seeds testing low germ. What can I do about it? I didn't know. But I know how to read. I know how to look at the world literature, read scientific literature, and I know how to do experiments. And I figured it out. Crop decline, main limiting factors. I work with Dr. Matichenkov, who's a Russian scientist. He worked in Florida Citrus Commission and AFAS, to determine what's causing decline in citrus. This is like 20 some years ago, more than 20 years. And so it boiled down to what he said, activated humic acids, including fulvic acids, and silica, monosilicic acid. So the two main limiting factors, and I found out it was the same in these other tropical crops I was working with, like mac nuts and other stuff. So humic substances and silica, or monosilicic acid. So based on soil and plant testing, specific trace elements may be needed. Low soil organic matter and very low microbial levels with bacteria dominating need to be reversed to fungal dominance. Soil compaction, you need to deal with that, plow pan, etc. There are certain micronutrients that aren't important for plants, but they are for microbes. And we test for them and we provide them in our product called TM7, which is 7 crucial trace elements that's usually missing, and they're in the right ratio too. So the BioAg program for grain and seed crops in northern states and Canada. Seed treatment using microbial products with organic biostimulants and trace elements. Apply humics with a planter or via irrigation or pre-plant banding. Mixing or coating urea fertilizer with full humic powder. We just coat, it's black, it sticks to the urea nicely. And in China, they call that black urea. Black urea. Mixing or coating, yeah. Plant or petiole testing. I'm a real proponent of testing, sap testing, nutrient soil testing with lysimeters and all that to monitor what's going on. And then dealing with it. We test, and then the next day we'd go out either with ground equipment or aviation and spray foliars. I'm also a pilot, but I didn't do the spraying much, but I was mostly going out to farms all over the Northwest, landing in their field, taking the samples, going back to the lab, making the recommendation, then the spray pilots would come out. So we'd get a couple thousand acres the next day done. So then we're using cytokinins, and that's the plant growth regulators you find in kelp. And then decay system management. So what's going to happen with all that crop residue? We want it to turn to humic substances, not vinegar. Farmers would come to me and they say, Bob, I plow up my ground and the straw is still there from 2 years ago and it smells like vinegar. Okay, you need air, you need oxygen in the soil, you need to change your tillage methods. We use an activator. We use our product called Cytoplus, which is a growth stimulator, cytokinins, trace elements, humic substances, to speed up the decay residue so it goes the right direction to humic substances. So that's the basic thing. Seed treatment, pre-plant, treating urea if you're going to use it. Soil test, whether you're going to use a trace element product, and then monitoring through the growing season, and then managing the decay systems when you're done. That's it, folks. What worked in Alberta? I worked with growers in Alberta that were growing canola and wheat, and what worked was the TM7. That's the product with the humic acid plus the 7 micronutrients and sulfur. It wasn't registered. We'd ship it up here in a plain brown paper bag. It's registered now, so you can read the label. Okay. So it contains these elements, like I say, some of them to help the microbes: cobalt, molybdenum, and then. So why would you use it? Let's get back to some numbers, I guess, or dollars and cents. Okay, this shows the different yields based on the amounts of humic substances applied. It doesn't show up very good, but you see the top ones there: panic grass and buckwheat, are very responsive. And then there's corn, wheat, barley, tomatoes, cauliflower. All those plants, cauliflower, broccoli, cabbage, really, really responsive. And then you have all the other crops here, beets, and so that's the average of all crops is the yield increase you're going to get if you do it right. Sunflowers, it's a funny thing, the oil crops like sunflower, soybeans have less response to pure humic acid. If you use the trace element form, then they have a very big response. So you're bottom line, it's okay. Crops that produce oils have less of a response than crops that produce sugars. And like fruit crops are really responsive, like apples, citrus, things like that. They produce a lot of sugars. And the quality factors: vitamin C, beta carotene, the vitamins, proteins, etc., go up. The sugars. But it varies. See, tomatoes has a higher than sunflowers. That's what I'm saying. Sweet things have a higher response than oily things. And how much you use? Well, you have to do a lot of field testing. Okay, so on this side, we have the percent over control of increase in yield. Now, this was done on carrots, which are a sugar, they produce a lot of sugars, okay. So here's the rates used of the soluble humic substances, different rates, different concentrations, and you get into the signal of the energy signals involved, and the concentration is crucial to how that works. A Russian physicist explained this to me, but I can't explain it because it doesn't sink in. I'm not a physicist. I know about concentrations. So there's an optimum concentration. There it is, the dot at the top. If you go beyond that, add more, beyond the optimum, what happens? Starts going the other way on you. And if you add a ridiculously high amount, it goes back to control. There is no benefit. So there's a sweet spot that you have to hit. Now, if you go down here at these lower rates, you're just wasting your money, the cost of application. If you hit the sweet spot up there, which is about 0.15% or 30 kilograms per hectare, that's a sweet spot. Beyond that, no benefit. Much beyond that goes negative on you. That's important to know that. It's very important to know what the concentration and how much. And we know that. These other companies, they're just shooting from the hip, So these are the optimum rates that were accepted worldwide, 67 and 375 grams. First of all, you got to know what the actual content of the product is in humic and fulvic. If you don't know that, you don't know what rates to use based on the data. So you have to have a guaranteed analysis based on a credible test method to start. Otherwise, you're just shooting from the hip. So our products are certified by both OMRI for organic use and by Humic Product Trade Association and by the state, the different states like California, Oregon, even Canada. Which, the nice thing about Canada, we only have to register it once for the whole country of Canada. In the US, it's every state. Yeah. So this is what biostimulants are. Humic acid, seaweed extracts, protein hydrosalate, which is amino acids, glycine, betaine, and growth-promoting rhizobacteria. Azospirillum for winter wheat, improves root morphogenesis. And we sell these microbial products. We produce some really interesting stuff. So we're into the whole, matter of fact, we're in the whole range here. We provide the whole range and more. But we have the living stuff, the microbes, and we have the dead stuff. And we have the mineral stuff. That's going to work, Vic? Now this is actual testing on our test farm in the Willamette Valley.
Here we are where we used dry humate pre-plant, quite a bit of it, sprayed out here pre-plant. So here's the rye. You can see here's my stick. So this was put on here last year, showing the long-term carryover benefit of humic substances. And then here's where we didn't put it. So which crop would you have? Which would you rather do? Of course, we'll see a big, gigantic difference when the yield at harvest.
And we did.
Okay, let me just walk over. Here's without humate. We're not worried about organic matter, just chemical fertilizers, salts. We're good. That's the mentality. Okay. So here's large applications, optimal applications of humic, full humics. That's humic, fulvic, the whole spectrum right here. Here's without. Here's with. So, you don't believe your own eyes, fine, there's nothing I can do.
Okay, next one.
So this
And this is rye for seed. We grow seed
and
in
BioAg
in Willamette Valley.
And we're gonna go over to the field over on the other side of the road, and had none of that. And it was planted with rye the same day, and you can see the difference. So here's my indicator stick. You can see the thickness of the rye, and it extends pretty uniformly in this particular test plot. But you can see the leaf width. That's the key. In grain, barley, and corn, you'll see a wider leaf blade.
In a blue color, a blue tint on the leaf.
Now here's where they damaged the leaf with herbicide spray. You can see the burn. Because they insisted on spraying this unnecessarily. Okay, so here we are in another part of the field. And this is the untreated, without the mycorrhiza, without the full power of Fulvic on rye. Same day. And we walk over here.
One of the ag planes flying by.
Untreated. Boom. Here we are in the trees. And you can see a difference in the color. Bigger. There's spray plane going over, military rescue.
Okay. Yeah, our farm is also an airstrip. It's airports on. But anyway, so organic rye seed crop at our research farm. We're up to harvest there. There's Vic and.
Here we are in the Faust Bio-Ag farm. This is a crop of rye, biologically grown rye, using Cyto Plus foliar and BAM. You can see the size of the field.
And I'm about 6 feet 4 inches tall. This is about 6 feet tall. When we sprayed.
It's a good 6 feet tall where we sprayed.
That's fall planted rye.
It goes down a little bit.
Here it's much shorter.
Thinner stalks.
Yeah, thinner stalk. And you can see the beginnings of lodging, where it's kind of falling and tangling a little bit. The heads aren't as big, the grain is smaller. Oh yeah, the heads are definitely smaller. Go over there and grab a head and we'll compare the heads.
A couple of heads up here.
Yeah.
I should have walked a little bit faster.
Yeah, I know.
So yeah, this
Now we're getting down to the wire here.
ground had Humic supplies and coming off of a vetch cover crop.
Vetch cover crop. Keep that in mind.
So here's the difference.
Yeah. So.
It's not just the height. You don't get paid for that. It's the number of grains per head and the size of the head, and you can see the big difference. Big difference.
Okay, we're done. Okay, well, in the Willamette Valley, we grow seed. It's a seed growing place. We grow all this grass seed. This is tall fescue on my property. The neighbors is the not so good. This is just spraying a liquid humic acid on that grass, on that fescue. They plant it this way because it's seed production. You see, they don't want a solid, like, it's a different ballgame, okay? So here's an aerial picture of the strip we sprayed with ground equipment versus the neighbors, the rest of the field. Here, this is our factories where we make our products, and this is our runway. And then, so big difference there. Okay, so here we go. Why do these things work for humans and animals? Well, because these different functional groups, mainly the aromatics, which are the polyphenols, tannins, activate this pathway, the TGF-beta pathway. And that switches on, that's the best way I can say it, switches on resistance and growth regulator response in the cell, and the plant is cells. Okay, so the whole thing goes. It works in the nucleus of the cell, switches stuff on, and then you also have the P38 MAPK pathway, and it goes through all these different steps. Plant gets infection, this happens, that happens, this happens, and we end up with pathogen resistance. The plants make their own plant growth regulators. You don't need to buy them. You just need to turn it on. Okay, so I don't have much time to go here, but on the longevity effects, using indicator animals, which is Heterohabilis. It's a kind of transparent nematode that has a specific life expectancy. So when we use our products at different rates, we can measure the longevity of that organism. And so it's based on that activation of the TGF-beta signaling and the insulin-like signaling and modulation and blah, blah, blah. And so the end result is it extends life expectancy and longevity. And it doesn't matter what cell or who's got that cell. It just, for lab work, we use this little nematode, and they use it in medical research because it gets through its life cycle quick. So that's the human and animal health benefits, not just plant benefits. And so you can find out about the human health benefits from my partner, who's an MD and a biophysicist, at monolifelab.com. And that's about for everybody in here, and everybody's crop and everybody's animals. And okay, I made the time limit. So human and livestock supplements made by BioAg, available from Monolife Lab, Dr. Elizabeth Wanek, MD, and me. And Monolife Lab, there's the products. You can go online, you can see it, all my YouTubes and stuff like that. Why snake oil medicines work, okay? And so here's BioAg, our company for agriculture, and these are the products. The mycorrhizal fungi, the trace element biofertilizers, the full power fulvic and the humic. And that's what we do since 1983. I'm still around. These competitors aren't. It must work, right? Okay, there it is. That's it. I'm out of time. I could go on all day, but.